expression of the aquarium protein. So, my introduction. The GFP is a green fluorescent protein that is extracted from a jellyfish, and this gene basically makes things glow. Breast cancer is a type of cancer that's really common and divides really quickly. And recombinant DNA is when two foreign DNA come together, and it's what they kind of use to make medicines and stuff like that. The purpose of this experiment is to find out whether or not the insertion of the green fluorescent protein into breast cancer cells will make the cells express green fluorescence. If this science is used, then it can help doctors find cancer cells within a person's body quicker so they stand out. Hypothesis: if the green fluorescent protein is extracted from the jellyfish, then the cancer and inserted into a cancer cell, then the cancer cell will emit the green fluorescent protein or light. This is because when it's put under a blue light, when bound to calcium, the GFP absorbs the light and turns it into green light. And if this gene is put into the cancer cell, then the cell will divide with it, and the divisions will when it divides, it'll have the protein and will express it. List is an incubator, blue LED light, microscope, and tissue hood, culture hood, and a freezer to put cancer cells in. Materials list is cancer cells, green fluorescent protein, petri dishes, expression vectors, alcohol, paper towels. Always wear goggles. Don't eat any of the things, I guess. And wash your hands before leaving the lab. The procedure is basically, well, you have to clean the hood first, make sure it's sterilized, and then you kind of extract the DNA from the jellyfish, and then you have to culture breast cancer cells, that is, and then you cut out the piece of DNA you want from the jellyfish with a restriction enzyme, and when you get that piece of DNA, you put it into an expression vector, which is a vehicle for moving DNA from one place to another, and expression vectors are especially used for when proteins want to be shown. So, and then you use DNA ligase to put the protein into the vector, and then you put the vector directly into the nucleus of the cancer cell, and then you have to put cells into the incubator to let them sit and kind of repopulate and divide, and then you take them out and put them under a blue LED light to show it and to see if you have glowing cells. The variables. Independent variable is the GFP put into the protein. Dependent variable is whether or not they emit a green light. Control is the cells without the protein. Control variable is temperature, amount of DNA put into the cancer